So today we're going to be talking about the cell cycle, and I have my notes here with me to give a good explanation. So most cells remain in the interphase and rarely divide, and there's 5 phases. So the interphase consists of G1, S, and G2. And then you have the M phase, which consists of mitosis and cytokinesis. So interphase is that period where they are not dividing. And so within that, you have the G1, which is the normal physiology of a specific cell type. And then prepping for cell division would be the S phase, which is a genome duplication. And when that happens, then it goes to G2 phase, and that's the RNA synthase, is where the RNA synthase halts. And basically, they wait until they get a signal to go to the M phase. So the M phase, like I said before, consists of mitosis and cytokinesis. So mitosis produces daughter cells identical to mother cells, which is binary fission. And this happens in somatic cells. So the steps, you could say it's called karyokinesis, which consists of prophase, which is chromosomes visibly condense, spindle forms from centrioles forming an aster, and nuclear envelope dissolves. Then you have the prometaphase, is a sister chromatid attaches to a spindle by a kinetochore, and the chromatids move to the middle of the cell. Then you have metaphase, which is the chromosomes align at the center. Then you have anaphase, which is the chromatids separate via microtubules. Then you have telophase, where the spindles dissolve, nuclear envelope reforms, and chromosomes decondense, and cytokinesis begin. Cytokinesis is separating cytoplasmal material, and you end up with 2 daughter cells, and this happens in human. In animals, the cleavage first forms, and in plant cells is the cell plate. So then you also have something called meiosis, and there's differences between them. Meiosis produces 4 cells with half the chromosomes as a mother cell. It produces gametes, which in humans is the sperm and the egg, and in plants would be the pollen. Meiosis goes through 3 divisions, meiosis 1 and meiosis 2, and end up with half of the number of chromosomes, and that is called reduction division. So reduction division of a diploid germline cells, which is 2N, produces haploid germ cells, which is 1N, which matures into gametes. Then the fusion of gametes with fertilization produces a diploid cycle, or the baby, you can see. So a difference between mitosis and meiosis would be, so they have one round of cellular material duplication, or the S phase, but meiosis has 2 rounds of karyokinesis and cytokinesis, while mitosis only goes through 1. So talking about meiosis 1 and meiosis 2 now. So the biggest difference come from the prophase 1 and anaphase in meiosis, and that's the biggest difference between mitosis and meiosis. So in prophase 1 in meiosis, when the crossover occurs between homologous chromosome, and then does dissociating link genes. As well, the trachea gather at the synapses, and basically, instead of in mitosis, they have instead of forming the homologous chromatids, they form tetrads. Then in anaphase 1 in meiosis, homologous chromosomes, which is reduction. You also have the metaphase 1, which then goes the 3 sets of tetrads, and then kinetochores attach, kinetochores attach, and then the centromeres intact as well. Then you have the telophase, and that means 2 haploid cells, sister chromatids attach, and independent assortment. So independent assortment is like an example would be, you have your red would be the mother and blue would be the father. It won't automatically go all fathers and all mothers. It would just go by itself. So it could be a mixture of both. It won't separate by the mother and father. The chromosomes. So then you also have meiosis 2, and that does not go through the S phase between meiosis 1 and meiosis 2. And the M phase is similar to mitosis, the same thing, but except it doesn't go through the S phase. And anaphase 2 is where the sister chromatids separate.